and welcome everyone to this podcast for cell biology. In this podcast, we're going to focus on tissue maintenance and renewal. And we're going to spend a lot of our time talking about stem cells in this larger discussion. Up to now, we've talked about how a specialized cell, so a cell that we'll say is differentiated, can divide. We've talked about how a differentiated cell can go through apoptosis and die. We've briefly talked about how a cell can grow. We've talked about how that cell can simply just survive without dividing, dying, or growing. But what we haven't talked about is where does this differentiated cell come from? And how do we maintain these differentiated cells? Once they die, how are they replenished? To begin this discussion, let's look at a zygote and consider one of the most fascinating questions in all of biology, and that is how you take this one cell fertilized egg, the zygote, and ultimately form this fully functional organism. In this case, let's talk about humans. You will have 10 trillion cells. The fascinating thing about this one cell embryo and this 10 trillion cell organism is that the genomes are identical. Every single cell in this human has the same genome that this one cell zygote has, minus of course any mutation. To remind you, this cell will go through several cell divisions, and eventually it'll form something called the blastocyst, and then it will form the gastrula. I'm not going to try to draw this, but I want to mention it because the gastrula is the stage at which tissues begin to form. Cells begin to differentiate at this blastocyst stage, but at the gastrula stage, they further differentiate, and their destiny of becoming certain kinds of tissues is set. This gastrula will then go through many more cell divisions before forming this fully formed human being. And so we call this whole process, at the cellular level, we call it differentiation. How do these cells differentiate? And by and large, and we'll talk more about this, they differentiate by having a change in the gene expression pattern based upon cues they get from the environment. Now let's talk a little bit more about these tissues. Now, I mentioned they begin to form here, but let's think about fully specialized tissues in the fully formed organism. They will have several kinds of cells, or types of cells would be another way of saying it. Each type of cell must remain unique. Meaning if you're talking about a pancreas and you have a beta cell, that beta cell has to remain a beta cell. But it must also coexist with other cells. Not just coexist, but in many cases function with other cells in order to maintain the specialized state of that tissue. These different types of cells that make up the specialized tissue are continuously dying and being replaced. And they do all this while maintaining the tissue's organization and stability. Next, I want to talk about the three factors which help maintain this stability and this organization. Because if you didn't maintain this organization and stability, the different tissues that make up this organism would cease to function and the organism would cease to exist. So it's important to maintain their stability. So let's talk about that next. So the factors that maintain tissue stability. The first is cell communication. Cells within the tissue monitor and respond to the environment, mainly through signals that they receive from neighboring cells. Through this cell communication, cells will divide, grow, survive, and die when needed to do so. The second factor is something we'll call selective cell adhesion. And this is accomplished through the use of selective cadherins. Remember, these are the ones that are used in those adherens junctions to help bind neighboring cells. So these selective cadherins help hold cells of the same type together so they can function together. You don't want every cell within a tissue binding to each other, because you need to maintain that organization and stability, and binding everything together just creates this glob of unuseful cells. Now, sometimes there is selective attachment to cells other types or the extracellular matrix. For instance, the cells of our epidermis that coat the outside of our skin will adhere to each other like so. And there's probably multiple layers here, so let me quickly draw this in here. So remember, this was the epidermis. Now, they also bind to cells of the dermis, which are going to be below it. And there will be multiple layers here as well. So the cells of the dermis use selective cadherins to bind to each other, but some of these cells of the dermis must bind to cells of the epidermis, so that these two layers of cells in this tissue can work together and communicate with each other. Without cell adhesion, cells would mix together and be functionally useless. The third factor to help maintain this stability involves something we call cell memory. Your book doesn't use this term, but the term that best describes this is something called epigenetics. And what we mean by cell memory or epigenetics is that the gene expression pattern in a specialized cell will remember pattern when it divides. Sometimes it's when that specialized cell divides, or when that precursor of that specialized cell divides. But the point is that a certain gene expression pattern has been set within that cell, so that every time it produces a new cell, it knows which genes to express and which genes not to express. That helps maintain this stability. You don't have to reprogram the cell every time you produce a new one. Some patterns of expression, maybe even many, are set during early embryogenesis, which is pretty amazing that at an early stage like gastrulation, where the embryo is just forming, some of those cells set a pattern that will be remembered for the next 70 to 100 years. Next, I want to talk about cell turnover rates. We've mentioned similar things before, but let's just remind ourselves of what we mean by this. Some cells, like a neuron, usually last a lifetime. They don't typically go under apoptosis. Some do, but typically they don't. And so once you have a neuron, it doesn't turn over. Other cells, like bone cells, have a turnover rate of 10 years. So it's a pretty slow process, but your bones do turn over. Then you have things like red blood cells, which turn over every 120 days. And then finally you have intestinal epithelial cells. And they turn over every 3 to 6 days. So let's spend a little bit of time talking about, once these cells die and are lost, how are they replenished? And to begin that discussion, let's define terminally differentiated cells. These are cells that have reached their final specialization. They typically don't divide to make new ones. Your book uses this phrase which I like. They say they have reached their dead end of their developmental pathway. They're not going to specialize anymore. They are what they are until they die. So once these terminally differentiated cells have reached their end and they go through apoptosis, how do we replace them? How do we replace a bone cell once it's been lost, a red blood cell once it's been lost, an intestinal epithelial cell once it's been lost? So let's talk about that next. To begin this, let's talk about stem cells. And for right now, I'm going to call this a somatic stem cell. We'll talk about these in a little bit more detail in just a moment. This is what is often referred to as an adult stem cell, but this is a better term for it, and I'll explain that in a moment. This stem cell can do one of a few options. This stem cell, when it divides, can do one of a few options. It can continue to be a stem cell and replenish the population of these somatic stem cells, so it does not differentiate. So I'm going to write no differentiation. Or it can differentiate. And depending on which pathway you're talking about, it can become several intermediate cell types. But what we're going to call these here is a population of proliferating precursor cells. They left this somatic stem cell nature behind, and now they are in a pathway to become differentiated. Now this population of proliferating precursor cells can be quite large, but this is the source of cells that will be used through several rounds of cell differentiation to produce a terminally differentiated cell. A terminally differentiated cell. Now I drew it looking quite different, and it should be noted that as you go from somatic stem cell to your terminally differentiated cell, not only is there a change in pattern of gene expression, but the cell shape will also change, as well as this cell function. So let's write those three things down. So as we go through this process, gene expression changes. Some genes are turned on, some are turned off. Cell shape changes. And ultimately, cell function changes. The whole purpose of this elaborate pathway is to produce a cell that has a fully differentiated function. I should also mention that cells do not typically go backwards. Differentiation is usually unidirectional, from the zygote to a terminally differentiated cell in an adult. Now, there are cases, in fact lots of cases, in cancer where differentiation does go backwards. And we'll talk about that in the next podcast on cancer. Now, one other thing I want to mention here is that these somatic stem cells usually can follow multiple paths of specialization. So this first path produced one cell type. Maybe this somatic stem cell could have produced multiple kinds. So for instance, if this stem cell were a special kind of stem cell called a hematopoietic stem cell, it could become a whole host of different kinds of specialized cells or terminally differentiated cells. It could become a T cell, a B cell, neutrophils, macrophages, and other kinds of white blood cells. It could also become a red blood cell. Which one it becomes is all dependent upon what signal this somatic stem cell receives before it differentiates. However, its specialization is limited. In this example, it's limited to white blood cells or red blood cells. It can't become a pancreatic cell. So somatic stem cells have a limited ability to specialize. Still pretty powerful, but they can't become anything. Now some stem cells have a greater ability to specialize. And let's talk about the three kinds of stem cells. Okay, so let's go ahead and talk about our three types of stem cells. I should say naturally occurring stem cells. We'll talk about some induced stem cells later. But as we do this, let's consider embryogenesis to talk about these three types of stem cells. Let's begin with this one cell zygote, and it will divide and form 2 cells, and then again divides to form 4 cells, and then it'll form this multi-celled embryo. It will then form a blastocyst, and the blastocyst contains largely 2 cell types. The cells here in the middle are called the inner cell mass, and the cells on the outside surrounding the inner cell mass are called the trophoblast cells. This blastocyst, I should write blastocyst so we don't forget what that is, blastocyst. Okay, these blastocyst cells will then divide to form the gastrula, and then ultimately go on to form a human being, in this case. It could be any organism, but we're going to focus on humans here. These early cells, before they become a blastocyst, are what we call totipotent stem cells. They can become any body cell. They could ultimately become any cell in, well, first this inner cell mass, but ultimately in the fully formed individual. They can also become any, what we call extra embryonic cell. These are the cells that make up the amnion, the placenta, something called a yolk sac, and something called an enlantois. I would like you to remember that the extra embryonic cells make up the amnion and the placenta. So they have total potential, totipotent, to become any cell, either in the body or the extra embryonic cells. I should also point out that these trophoblast cells, these are the ones that will become the extra embryonic cells, and the inner cell mass cells here have the potential to become any cell of the ultimate organism here. And so these inner cell mass cells are what we call pluripotent stem cells. These are what are often referred to as embryonic stem cells. They can become any body cell. As it begins to form tissues in the gastrula stage and beyond, these cells can become any of those cells found within tissues. They cannot, pluripotent stem cells cannot become the placenta, the amnion, yolk sac, or enlantois. So they have begun to differentiate at this state. The last type of stem cell I'll have to draw down here, and these are called somatic, or you could call them multipotent stem cells. These begin to form in the gastrula stage, and ultimately throughout embryo development and fetal development and in the adult. These are often called adult stem cells. I don't like this term because these stem cells, these somatic stem cells, begin to form early in embryonic development, far before those cells become an adult, the way we would typically identify adult cells. So to call them adult stem cells is a little misleading, I think. But somatic stem cells or multipotent stem cells makes sense. We can think of these somatic stem cells as having limited ability to form different cell types. Like we saw on the last whiteboard, we had the hematopoietic stem cell. That is a somatic stem cell. And as I drew, it could become many different kinds of cells within the blood cell lineage. But that's it. It couldn't become any other cells, any other tissues. Now I want to spend a little bit of time talking about these pluripotent stem cells. And I want to talk about some uses of pluripotent stem cells. So if you were to take a blastocyst here with its inner mass cells here, and you removed those inner mass cells and placed them in a petri dish, those cells would grow. In fact, they'd grow indefinitely as long as you gave them the right nutrition as an external source of embryonic stem cells. You could take these cells and you could give them different signals, and you could make, experimentally in the lab, all kinds of cells. You could make fat cells, neurons, red blood cells, different kinds of muscle cells, even different kinds of glial cells. Now the goal someday might be to use these embryonic stem cells to treat disorders where nerve cells have been damaged, like in Parkinson's disease, or muscle cells have been damaged, like in muscular dystrophy. One of the problems, of course, is rejection. This source of embryonic stem cells didn't originally come from the person being treated here, so there is a possibility of the body rejecting those stem cells. So let's write here, disease treatment with a question mark. That's where we're headed towards. Now we know that these are, in fact, embryonic stem cells, meaning they function as embryonic stem cells, because you could take these cells and you could place them in a developing embryo, and these stem cells would incorporate into the new embryo. So if we have our embryonic development here, we place in our embryonic cell here, it's the only one, or maybe there's a couple here, into this developing embryo that's not related to that one. As this embryo develops, it will contain a mixture, different tissues will contain a mixture of the original cells with some of the transplanted stem cells. In a sense, remember that they're an embryonic stem cell, and if you place them with other embryonic stem cells, they can receive signals to continue their embryological developmental pathway. Now I want to talk about these two terms, reproductive and therapeutic cloning. Often I hear in the media these two terms used interchangeably, but they're very different. So let's talk about them and talk about how they could potentially be used. In both cases, you take an oocyte that contains a nucleus. The nucleus is removed, so you have an enucleated oocyte. At this point, this oocyte could not be fertilized and become an embryo. But what they can do is they can take DNA from a terminally differentiated cell and place it into this enucleated cell, or oocyte. Now remember, this is a diploid 2N nucleus because it came from a terminally differentiated cell. In essence, what has happened is you've created an oocyte that genetically resembles a fertilized egg because it contains 2 copies of each chromosome. The only difference is it came from one cell. This oocyte that now contains this 2N nucleus will be stimulated to form an embryo, and we'll draw this blastocyst stage here. Now remember, each of these cells will contain the nucleus of the donor nucleus. Now in reproductive cloning, this blastocyst is placed into a surrogate mother and the progeny is allowed to form, and a cloned organism is formed. This is how Dolly, if you remember or have heard about, the first mammal that was cloned in this way, Dolly was cloned. She was a sheep that was cloned in England. Now there's a lot of ethical concerns and also scientific roadblocks that would prevent this occurring for humans, to clone a human being. To get around some of the scientific and ethical dilemmas associated with cloning humans is that we can take these cells and we can grow them in a petri dish. And these cells will divide, and I'm going to draw these in blue to signify that they're going to resemble the donor nucleus more so than the oocyte. So these are cloned embryonic stem cells. They no longer look like this terminally differentiated cell. They've been reprogrammed, and we can grow them as such in a petri dish outside of the body. Ideally, then, these cells can be used for therapeutic purposes to potentially in the future treat certain diseases. I should have wrote therapeutic cloning on this line. Okay, as a quick review, reproductive cloning would produce a whole organism that would resemble the original organism that donated the nucleus, and therapeutic cloning here still produces the embryo but not the whole organism, just the cells. Now, using this approach still relies on using an oocyte and still relies on forming an embryo. This next technology I'm going to talk about eliminates the need for these two steps here. Okay, this last piece of technology I want to talk about is called induced pluripotent stem cells. We often see this reported as iPS cells, or sometimes iPSCs. And using this technology, one would take cells from the donor patient, so maybe from his or her arm, and so this would be an epithelial cell, a terminally differentiated epithelial cell with its diploid nucleus. So the next thing they can do is they will take this terminally differentiated cell and they will overexpress 3 genes. They are OCT34, SOX2, and KLF4. These genes are transcription factors, and when we overexpress these 3 genes, it causes this terminally differentiated cell to convert back to a stem cell. So this is what we call an induced pluripotent stem cell. We have forced a terminally differentiated cell to become, in essence, a pluripotent stem cell. We can now take these cells and grow them in a petri dish, and they will behave just like an embryonic stem cell. We can take these cells, just like we saw before, and specialize them into terminally differentiated cells, depending on what kind of signals we give them. The idea is that we could use these for a therapeutic approach. An advantage to this process is you're not using an oocyte, you're not creating an embryo, and there's less of a fear of rejection because you're taking it directly from the patient that needs, or potentially needs, these stem cells. Now, they're not perfect. They don't have the same kind of potential as a true pluripotent stem cell, but they could still be used potentially to form new differentiated cells and possibly in the future use them for therapeutic purposes. Okay, that's all I have for this podcast. If you have any questions at all, let me know. If not, I'll see you in class. Bye.